Pathophysiology of AIDS, Acquired Immunodeficiency Syndrome. AIDS is a chronic, potentially life-threatening condition caused by the Human Immunodeficiency Virus, HIV. After getting infected with HIV virus, it enters the bloodstream and attacks the host immune system, mainly T cells. The virus, with the help of its surface proteins, glycoprotein 120, GP120, attaches to the receptor present on T cells called CD4 receptor. This attachment is further strengthened by a co-receptor called CCR5 present on the surface. This attachment causes a conformational change and fuses the cell walls of both the virus and the T cell. This fusion causes release of the virus components into the cytoplasm of the cell, which include two single-stranded RNA, three enzymes, reverse transcriptase, protease, and integrase. The reverse transcriptase enzyme makes another copy of viral RNA and leads to formation of double-stranded DNA called proviral DNA. The proviral DNA then enters the nucleus of the T cell. Then the integrase enzyme makes a cut in the host DNA, integrates the proviral DNA into the host's DNA. This proviral DNA is transcribed to form mRNA. With the help of enzyme protease, this mRNA directs the synthesis of HIV proteins. These HIV proteins, assembled along with the viral RNA and replication enzymes to form immature HIV virions at the host cell inner membrane and budded from the cell surface within a new envelope. Each host cell may produce thousands of immature virions, which are later converted into mature viruses. These viruses make holes in the T cells and eventually burst the cell to die and infect other host cells, and are also released into the blood circulation. Thus, HIV patients have very few T cells that help in maintaining immunity. This further makes the HIV patients prone to other opportunistic infections, which is combinedly called AIDS.